Drug discovery is a process by which new medications are discovered. When we talk about drug discovery, target identification and lead discovery are the two major steps of drug discovery. अब ये दोनों जो steps हैं, for that purpose we should have a clear understanding of the molecules which are involved in this process. As we have already covered that drug discovery के लिए जो molecules हम लोग use करते हैं, that are basically proteins. And as far as proteins are concerned, their structure is also very important. Now what is that structure and what is its importance for drug discovery, that will be covered during this module. If I go towards structure biology and importance of structure biology in drugs, what we would say that number one, we need to identify proteins with their ligand information. So ligand is basically our drug-like molecules which are very small and which are put with proteins to identify new drug targets. Then we have candidate drug screening. So candidate drug screening is basically the term used for screening many proteins to provide optimal or optimized protein structures or molecules. During 1950s, what we saw that there was an emerging trend towards structure biology thing and computational power of resources. So these two things gave us many new proteins and structural information of those protein type things. Most commonly used method of that time was X-ray crystallography. Today NMR stereoscopy is used. So why it is used and what is the advantage of this thing is that by using NMR, which is based on magnetic resonance technology, we can get many more molecules which can be used for drug discovery processes. Now what has happened is, if we look into details of biological data, we have roughly genome sequences of 240 organisms, but the total number of proteins with their ligand information is roughly 500. So as if we compare these two numbers, 240 organisms and only 500 proteins, that information is very small. So again, there is rapid work going on for identification of more proteins with their structural information. So what would be the advantage of getting this structure information? If we have a good complete understanding of structure information, it will help us to make good and druggable medicines for curing of diseases. Here I have given an example of protein structure. Basically, protein structure can be provided at two levels. On the left hand side of this figure, if we see the primary sequence which is written, this is basically a sequence of amino acids which are part of a protein. In this amino acid, we can see that these are only characters of different amino acids. If we want to get some information out of this primary sequence, it is very tough. So in order to get information about different type of data from a protein molecule, we use this 3D structure which is shown on the right hand side of the figure. What happens is, if we use this 3D structure, we can get other information such as where our molecule or drug will be combined, what would be the protein-protein interaction site, what would be the active site, and so on. So that is basically useful for converting of this information. Another thing to add here is that for proteins we have different level of information. For example, as shown in this figure also, at the lowest level we have sequence information. Then we have our binary structure, then we have our tertiary structure, and then we have quaternary structure. So there are different structures available, and as the structure level is increased, information about that particular structure also increases. That means that we can use that structure for good purposes and useful purposes. So here I have given an example of some drugs which have been discovered using structure-based approaches. Such as, if I talk about alovirin, it is basically a drug which is used for the curement of HIV/AIDS, and it is totally based on the structure of proteins information. Then other ones like capoten or so on, we have these different drugs which are used for different diseases such as hypertension, influenza, and so on. So basically what we want to conclude from here is that structural information is important when we want to design new drugs, because those drugs are based on protein information. And to understand proper structure of protein, it is useful to have its structural information. And for extracting that such structural information, we usually use X-ray crystallography or NMR spectroscopy. So based on these two things, we can get the structure of the proteins, and that structure is further used for drug discovery process.